Okay, now let's discuss about systematic sampling. Okay, systematic sampling is uh, is used when samples are taken at fixed intervals, usually along a line. Okay, this normally involves doing transects where a sampling line is set up across areas where there are clear environmental gradients. For example, you might use a transect to show the changes of plant species as you moved from grassland into woodland, or to investigate the effect on species composition of a chemical pollutant. There are two ways to do systematic sampling. First is line transect. The second one is using belt transect. Okay, so in this lecture, I will discuss on line transect. Okay, for this method, a transect line can be made using a nylon rope, raffia rope, or marked and numbered at half meter or 1 meter intervals, or any desired intervals necessary, all the way along its length. This is laid across the area you wish to study. A line transect is carried out by unrolling the transect line along the gradient identified. The species touching the line may be recorded along the whole length of the line as a continuous sampling. Alternatively, uh, the presence or absence of species of each marked point is recorded as a systematic sampling. The transect diagram here illustrates the type of data which is collected using a continuous line transect. This notes every individual which touches the line, so which means that across the line, so every single individual touches the rope, it will be counted and recorded. The data is displayed in the form of a diagram using symbols or for different species for this kind of study, which are drawn to scale. This is a useful way of being able to clearly visualize what changes are taking place along the line. So it enables patterns of zonation in species along the line to be picked out. You must be aware that the position line is very important, and it depends on the direction of the environmental gradient you wish to study. It should be thought about carefully before it is placed. You may otherwise end up without clear results because the line has been wrongly placed. It is likely that the transect line would be laid in the wrong area, and the results would be very confusing. Therefore, the topography of your study areas must be considered carefully. The selection of the areas must follow your study objective. Time is usually money, so it's worthwhile thinking about it before starting your sampling study. So why use line transects? Line transects are used when you wish to illustrate a particular gradient or linear pattern along which communities of plants change. They provide a good way of being able to clearly visualize the changes taking place along the line, depending on how detailed the line transect is. So they can usually accomplish fairly quickly. Line transects do not produce as much information on the relative densities of individual species as a belt transect would do. I will discuss about this later. A line transect tells you what is there, but gives limited information on how much of it is present. If detailed density information is required, then a belt transect must be carried out instead. Where there is a great deal of plant cover, continuous line transects are useful only over relatively short distances. If there are a great many plants touching the line, it is very time consuming to survey over long distances. It is also difficult to adequately display the data so that it can be shared with others. However, where individual organisms are sparsely distributed along the line, for example, in a desert or on a barren land, continuous line transects become workable over a longer distance. So it is important to bear in mind that the type of method in line transect which is most suitable will depend entirely on the habitat under investigation and on the purpose of the survey.